chapter 11 notes. This is going to go from karyotyping down to the end. Karyotype, one of our vocab words from last week, is nothing more than a graph or a diagram of the individual's chromosomes. So in humans, we would have 22 autosomes or body chromosomes, and then we would have our sex chromosomes because we have 46 pairs. When we map these out, this is where we can detect if we have any defects, any chromosomal abnormalities. Sometimes these abnormalities can cause problems. So here is a karyotype of a human. Now, for example, if it's a different species, it may not have 23 pairs, but we have 23 pairs of chromosomes, and this is also based on how big they are. Notice that your number 1 chromosome is larger than your number 22 chromosome. Also, look down here to the X and Y chromosome. So obviously this is the karyotype of a male because we have an X and Y chromosome there. But notice how much larger the X chromosome is than the Y. That's because it has a whole nother leg to it. You can remember that based on what the shape looks like of an X and a Y. So this is also another reason why males will show that recessive trait so much in sex-linked disorders. So if we have a number of chromosomes that is abnormal, usually what happens is a human embryo, if you have a big disconnect here, will usually miscarry naturally. The baby will not form to to the point where it can be birthed. There are some chromosomal abnormalities, however, that will upset the genetic balance and it will cause some defects, but the embryo will still develop and the baby will still be born. What are some causes that can give us some different chromosome numbers or abnormalities in the number of chromosomes or that type of thing? And what it can be is non-disjunction. This is where members of chromosomes, pairs, fail to separate. So when we talked about meiosis and the two parts where we have separation of chromosomes in meiosis 1 and meiosis 2, if in either one of those spots the chromosomes don't separate, you can have gametes that are not normal, which then when they join with other gametes can give us abnormal chromosomal things at the end. So if it occurs in meiosis 1, those homologous chromosomes that we talked about before, they do not separate. And what happens then, that would be all gametes have abnormal numbers. So every gamete that is created have abnormal numbers because you're never reducing the number as what happens in meiosis 1. During meiosis, what happens is you're multiplying them more in meiosis 1. During meiosis 2, those sister chromatins do not separate. And when that happens, I get 2 gametes that are normal and 2 that are abnormal. So you need to know that those abnormalities can occur. So here's a great picture of it. Here we have our homologous chromosomes. And in non-disjunction, you notice they do not separate. I should have a red on the left side one and a blue on the right, and it does not happen. In normal meiosis, it would happen. So then when I go down to reduce them, instead of having 1 chromosome in each one, I have 2, and then I have 2 that are blank. If you look over in the second side, everything seems to be pretty normal, except what happens is the non-junction occurs during meiosis 2, and those sister chromatins, or the X's, do not separate. And so they get 2 on one side and 1 on the other. And so then that can cause problems there. All right? Now, if either of these events occur, surviving offspring that result from these abnormal gametes will have abnormal karyotypes. So if the embryo does survive and the baby's born and we do a karyotype of them, obviously the karyotype is going to be abnormal. So let's look at some examples of that. Trisomy 21 is when we have non-disjunction occurs, and in chromosome 21, and the affected individuals have 3 chromosomes in 21 instead of 2. And what happens here, it leads to Down syndromes and the effects that Down syndromes can cause. This is what the karyotype looks like there. So if you go all the way down to number 21, you don't have a pair there, you have a tri. That's why I call it a trisomy 21. There's 3 of them, like our prefix tri. Another disorder is called Klinefelter syndrome. And this is when the males have an extra X, so it's XXY. And what happens here is they have sterility, which means that they cannot reproduce. You have breast enlargement, because the more X's you have, the more estrogen you have, though you have normal intelligence. But as the number of X's and Y's change and get more than just that XXY, you can get more disability in the mental aspect of the person. So that's Klinefelter's. And this is what the karyotype looks like. And if you go down here, first of all, you can tell that the Y is really small there, but you have 2 X's there when you look at the down bottom right where the sex chromosomes are. Another one is called Jacobs syndrome, and this is when we have an extra Y in males. It's an XYY. And these males are normal in terms of intelligence, that type of thing. They're usually taller and larger than average with Jacobs syndrome. We also have what's called a superwoman syndrome, or metafemales, and these guys have an extra X, so they get XXX. They're normal in terms of intelligence, that type of thing, but they are limited in their fertility. So they can be sterile in some cases or have very difficult times getting pregnant and carrying their babies to term. Turner syndrome is females that are lacking an X, so it's an XO. They're very short in stature, they're webbed necks, they're sterile, so they have no fertility. Poor development of secondary sex characteristics, so the development of breasts and that type of thing is very limited, but they do have normal intelligence. Here is a karyotype of that, and notice that's where you find that chromosome missing there. So those are some disorders that can come along with, and that is what's important about karyotyping, is that we are able to map those out. So hopefully you filled in your fill notes for this, and this is part of some of the stuff that you're going to have to study for your Chapter 11 test, and we will go over a bunch of the notes when I get back. So I will see you guys all tomorrow. Make sure you fill in your notes,